In this video, we continue to discuss the principles of model design with a case study that aims to model the growth of cancer cells. We also introduce principles of stability analysis for equilibrium points. In the model of bioreactor discussed in the previous module, module 2, we explicitly modeled the growth and loss of bacteria in a chamber. So these two terms here, the first term is the growth term, and the second term here is called loss term. Okay, both are proportional to the density of bacteria. So for the growth term, because the coefficient is positive, this positive sign in front of it, it indicates the growth, the increase of the bacteria over time. For the loss term, the sign in front of it is negative, so it indicates a decrease of the bacteria over time. That's why here R we call it production rate, and S we actually we call it the extraction rate, or the effluence rate. So we also provide a standard way to capture the saturation of the chamber with bacteria. So this was done by introducing the upper bound B on the density of bacteria inside the chamber. We also could carry capacity of the chamber, meaning that X, the density of bacteria will reach a maximum density of B. Okay, once X equal to B, so this term equals to 0. That means the bacteria, the density of bacteria will saturate. So the terms forming the model can be generalized to species or populations of organisms that grow according to a logistic factor and must survive in a closed environment with finite carrying capacity. Okay, so this means that we can use the same type of equations to describe other phenomena. Okay, for example, we can use the equations with similar structure to describe the proliferation of their E. coli bacteria, okay, in their living environment, such as the gut flora, okay, because their E. coli bacteria is found in the gut flora. Okay, so what has changed is here we use X1 as the density of the E. coli bacteria, and parameters R1, B1, and S1, which are specific to the problem, okay, specific to the E. coli bacteria, and typically are estimated from experimental data. So the point I want to make here is that the way we model the growth of bacteria in the bioreactor is actually a model template. So it's a class of models called growth models that apply to any physiological process involving growth with a logistic factor. So the interesting aspect of this class of model is that growth models such as the bioreactor can be easily expanded to describe the competition between two or more types of populations. For instance, let's say that in the same living environment, there are two types of bacteria. One is called the E. coli, we just described, the E. coli bacteria. The density is described, expressed as X1. Okay, suppose there's another type of bacteria, is P. originosa bacteria. So which are bacteria found in the skin flora. Okay, so these two types of bacteria are interesting because when placed in the same culture, Okay, they tend to fight each other for nutrients. And therefore, they are going to affect the proliferation of each other by stealing the nutrients from the other, okay, from the other organisms. So the question here is, how can we model the effect of the fight or the competition coming from the aeruginosa on the density of the E. coli bacteria? So our standard way to model competition between populations is to modify the loss term to account for the loss term here to account for the co-presence of both bacteria in the host or in the environment. So here, the product here, a product density, instead of S1 times X1, we change it to S1 times the product of X1 and X2. This indicates that the loss will increase. It's either there's a surplus of E. coli, okay, or that you can interpret this like when there's more E. coli, the dying rate of them will be higher. So that loss term is indicate that their dying rate is higher. And also the other case is when there's a lot of P. aeruginosa, because P. aeruginosa is their competitor. Okay, so which will deprive the E. coli bacteria of important nutrients. So that's why this term we introduced an X2 term here. Okay, that means X2 term when there's a more of the P. aeruginosa present in the environment, then there's a higher loss term. The loss term will be higher. Okay, for this equation describing the density of E. coli, which is X1. Okay, so using this multiplicative term here, we are expressing that the competition between E. coli and P. aeruginosa bacteria, they are competing for the same resource or same nutrients. So we can obviously do the same thing for the density of X2, which is the density of the P. aeruginosa. So we write this equation in the blue ink here. I'm going to just read here. The new term accounts for the fact that the amount of E. coli killed depends on the amount of P. aeruginosa in the same environment, and we must pair the equation with a model for the growth of the E. aeruginosa. That's the second equation for X2. So the parameters in this model are specific to the type of organisms involved, capture the rate of proliferation of each organism, and should be estimated from experimental data. All these parameters, S, R, and B, shall be estimated from experimental data. They're specific for each organism. So let's stress out one thing here is the use of this term, the use of this multiplicative term in both equations, okay, in both equations to express the loss of bacteria. Okay, this is a way to to model. Normally, we use this way to model competition between two or more populations in a given environment or a given host. So what shall we do now? Before we try to solve these two systems of two sets of equations, differential equations, just like what we did in module 2, the next step is to find the equilibrium point. Okay, how to find equilibrium point is we know that how to find equilibrium point. So it makes sense why we need to find the equilibrium point here, okay? Because we want to see how the equilibrium of each of the two populations changes, okay, because of the competition. So we should realize by now, we already did it for the last module, that computing the equilibrium point means that we setting the derivative to 0. We're setting the derivative to 0. Okay, setting derivative to 0. And solving, basically we're solving the right-hand side. Okay, now right-hand side is just the, we make them to be 0. So it's just is a set of two algebraic equations. In this problem, I think it's very easy to, just by looking at the two equations, it's very easy to see there's three, okay, to see several equilibrium point. So this first three are very easy to spot. So you can see 0, 0. What 0, 0 means? 0, 0 it just means really, it's just really trivial solution. It means there's no, there's either E. coli or P. originosa bacterials in the environment. None of them exist in the environment. Of course, none of them, nothing will happen. Okay. And the second one here means their X1 means the bacterial E. coli reach its maximum saturation density of B1. And then the P. originosa is 0. That means in the end, the E. coli triumph and by killing all the competitors of their P. originosa bacterial. Okay, and then the third equilibrium point is the opposite, is the P. originosa triumph. Okay, on the other, so that by killing all the E. coli bacteria. So all these equilibrium point are very sort of ideal scenarios. So basically, in the end, there's no competition going on because one of them is being killed or being just extinct. It is extinct. Okay, so it's more interesting to look for other equilibrium. Is there any other equilibrium? So then in order to calculate if there's any other equilibrium, we have to actually solve the algebraic equations. So let me see. Okay, solve these two algebraic equations. Finding the equilibrium is actually a very important exercise. And so let's try to think about how to calculate this by yourself. Okay, how to calculate this to solve this a set of two equations with two variables x1 and x2. Okay. So I'll show you how I did it. So I tried to get rid of the quadratic term. Okay, for the first equation I divide in x1. Okay, for the second equation I divide in x2. So now I get rid of their quadratic terms. Okay, so I just obtained like two linear equations in x1 and x2. So I rearrange it, it becomes this. So it becomes a linear equations. Okay, so how to solve this? Okay, we can use matrix operation to solve this, but I'm gonna just do it by, instead of use matrix, I'm just do it by eliminate terms. So I'm gonna eliminate x2. How to eliminate x2? So in order to eliminate x2, we need make coefficient in front of x2 to be, say, 1. So both of them to be 1, so that we call this their naked, so you can subtract them to get rid of x2. Okay, to make the coefficient in front of x2 to be 1. Okay. So in order to achieve that, I will divide the first equation by S1. Okay. And I will divide the second equation, or I will, the second equation, I will divide by R2 over B2. Okay, so to make sure that X2 is naked in both equations, that means the coefficient are 1. Okay, so I did so in order to get rid of X2 altogether. Okay, so now I have this equation. Okay, now I have this. You can see X2 are both equations. X2 has a coefficient of 1, so I can just subtract the two equations. So then it becomes a single equation for the x1. Then we can solve for x1. Okay. I'm going to show you how to do this in MATLAB. Okay, I'm going to show you how to do this in MATLAB. In MATLAB, we're going to use the, we can also, if you feel like that's too much work, that's too much hand calculations, you can just use symbolic calculations in MATLAB. You can define all the variables, all these variables and all the parameters, two variables, x1, x2, and all the other parameters, we call it the symbols. Okay, we define them as the symbols. And then we can just use this command with this function called solve. We put the both equations there. Okay. And notice that here use equal to indicate their equations. And then here indicate x1 and x2 are the two variables we're trying to solve. And then the equilibrium, the solution will be returned to solution x1 and solution x2. And then this is the output. These are the outputs when you run this this command. Okay, so okay, great. We can solve this, either by hand calculation or using MATLAB. When we do the hand calculations, and we actually can make the equation a little sort of more elegant. So we can call the sum of the parameter cluster, this cluster here to be 1 over beta 1. And then this cluster here, we call beta 2. You can see beta 1 and beta 2 actually are, if you invert this blue, the expression, you can see beta 1 and beta 2 is basically just describe the same thing. Okay, you change all this change all the 1s to 2 or 2s to 1, and beta 1 becomes beta 2. Okay, so why we do this is sort of just make the equations look more elegant. Okay, just look more elegant. So you can see that by doing this, so we get this equation here. Okay. So now of course, we can just subtract the two equations. By subtracting one, for example, the first one from the second one, we can have x2 removed. Okay, so I obtain a solution for just x1, expression for x1, equation for x1. Then I can solve we can solve the x1. We call in here, I call it x1 star. Okay, that's a solution for x1. Okay, so then you can replace you can just plug in the solution for x1 into either of these two equations, any of those two equations. Then you can obtain the solution for x2. Okay, which is reported here. So we report it as x2 star. So those are equilibrium point, the new equilibrium point. So by doing this sequence of steps, we found that another set of equilibrium point, which is x1 star and x2 star. So as we did in the last module, module 2, we need to determine whether this new set of equilibrium point are physiological relevant, okay, or meaningful, because we know x1, x2, they are physiologically, they means the density of the bacterial, and density of the bacteria and organisms has to be bigger than 0. Has to be equal, have to be greater than 0, or equal or greater than 0. Okay. So this equilibrium point is biologically relevant only if both the components are long-negative. So we can see in order for this two equilibrium of two densities, x1, x2, to be larger than 0, and then we have to satisfy either this set of sort of conditions, or this set of conditions. Okay. And so we have, and also we need to sort of consider whether the equilibrium point is stable. A stable means once you reach the equilibrium point, it will stay there forever. Okay, so if we, it can be shown, I will show you in the next, few slides. It can be shown that only if the top two conditions happen, the equilibrium point is stable, meaning that the densities x1, x2 will maintain indefinitely. So I will give, as I said, I will give a more formal definition of stability in a few moments. Okay, so now before doing that, before talking about stability, I'm going to ask what would happen if the two populations of bacteria do not compete for nutrients. Okay, if they do not compete for nutrients, for example, if they sort of consume different type of nutrients, or if there's abundance of nutrients present, so they don't have to compete for it. But then, however, they are coexist. If the two organisms coexist in the same host, they're going to compete for the limited space. So they're only going to compete for space, but not for nutrients. In this case, which is actually the kind of the scenario for, we are going to talk later about the cancer growth. Okay, in this case, the loss term should go back to the original form. Okay, should go back to the original form because bacteria of one population do not die nor are kicked out of the host or environment because of presence of the other population. However, the two populations of bacteria will still compete for the same limited space within the host. Okay, if the two populations were in the bioreactor, okay, the volume of the chamber would be finite. And therefore, for example, the E. coli bacteria would take space from the P. originosa bacteria and vice versa. So this can be observed. So this can be observed by observe that there's a cap on the density of bacteria on any species within the host. And this cap must be observed regardless of whether there are materials of just one population or two populations. Or accordingly, we can use a linear combination as reported in blue here to account for the competition for space between the two populations of bacteria. Okay, so notice that here, the B1 and B2 have very specific meanings, okay? So they are the maximum density that X1 and X2 would achieve respectively if they were alone. Okay, so they are specific to the population. Okay, when I look at the saturation B1, I'm seeing that this saturation would be reached when X1 is paired with the presence of X2, according to a coefficient I call alpha 1. So the fact that we are using a coefficient alpha 1, which might be different than 1, accounts for the different sort of the nature of bacteria, like different geometrical nature, for example, the size of their bacteria. So I'm going to give you an example here, okay? So I'm going to give you an example to explain the notion of this coefficient alpha, okay, alpha 1 here. Let's say that every unit of P originosa is twice in size in unit. Twice in size a unit of E. coli bacteria, okay? And therefore, the density of P. originosa will count as twice the density of E. coli in reaching the maximum capacity B1. Hence, we shall expect that alpha 1 in the first equation is 2. Okay, So I can do the same type of argument for the growth of P originals that enrich in the saturation of B2 for the second equation. So the point here is that the saturation depends on the type of bacterial. We are dealing with and the space we have to account for the fact that certain space is stolen for one population by the other population. We introduce the linear term, okay, this linear term, okay, linear term with coefficient alpha that accounts for the geometrical or the size differences between the two organisms that form the populations. So altogether, we have discussed two types of models so far. The models for logistic growth with competition and without competition for nutrients. So these models are, we call template models and apply to any type of physiological process that involves the growth of organisms. So for instance, we are going to model the growth of cancer cells in a living environment or host by using the very same type of equations we discussed, showing in the slides here.